In this video, we discuss about the various steps involved in cloning using cosmid vectors. We know that for any cloning purpose, we need a vector and we need a DNA of our choice. So here we have taken a eukaryotic DNA and digested with a restriction enzyme which cleaves a rare restriction site so that we are going to generate a larger DNA fragments. That is number one. So now here as usual it is a plasmid vector. I told you this has a unique cos site. So a plasmid vector with a cos site, we know that it is called as a cosmid vector. It has got an origin of replication. There is an ampicillin resistant gene. And in these cosmid vectors, we don't have a polyclonal site. Instead of that, we have a restriction site strategically placed. So one such is BAMH1. I will discuss the significance of BAMH1 in cloning purpose, especially using cosmid vectors. After the generation of larger DNA fragments of our choice, so we take this plasmid or the cosmid vectors and we have to linearize. To begin with, we digest with BAMH1 itself. So we are going to have a linearized plasmid. So it has an origin of replication, there is an ampicillin resistant gene and there is a cos site. So we take this particular species, we mix with this particular species of DNA fragments, do the ligation reaction using the enzyme ligase. So now because of the ligation reaction, since it is a random, three possible species are possible. Number A, what are the possible chances that this particular DNA itself can link one after the other like a tandem repeat. So there is a cos site here, there is a cos site, there is a cos site, there is a cos site. So the most important for in vitro packaging is the presence of two cos sites. Then there is distance. For example, now this cos site, if the distance is not between 38 and 50, it is not going to get packaged. What happens this one, this, again the same problem. Suppose if there is a possibility that there is a two cos site like this in this species, and if that size comes between 38 to 50, there is a chance that this can get packaged during the in vitro packaging. That is number one. If the distance is not enough, then it is not going to get packaged. Now another one is going to be this DNA fragments can undergo a similar linear polymerization. Since there is no cos site, there is no way it is going to get packaged in. Then the other species which is required by us is going to be the one plasmid DNA here, another plasmid DNA here. This acts like a left arm of the cosmid vector. This acts like a right arm of the cosmid vector. In between is the DNA of our choice. So now the distance between this and this falls in between 38 to 50 kilobase. This is the species which is required for especially in the in vitro packaging and subsequent into the infection. So this is the various steps. Now we will study the ligation using the actual vector, cosmid vector. All right. So now here I told you, so we have used the cosmid vector PJB8. Now the size of this cosmic vector is 5.4 kilobase, which is circular. All right. As I told you, this is an origin of replication. There is an ampicillin resistant gene and there is a cos site. And we have unique restriction site, but all these unique restriction sites are strategically placed. For example, now this is a cos site. On one side we have the SAL1. On the other side we need the HND3. Then there is a unique BAMH1. All right. So now this is the structure of a cosmid vector. And now this is the eukaryotic DNA. So what we do that we digest this eukaryotic DNA with SAU3A, SAU3A. Now this is a unique restriction enzyme and recognizes the unique restriction site. They are very rare. As a result, they do generate a large DNA fragment. All right, that is number one. And now here I told you that BAMH1 is very, very critical for cloning using cosmic vectors because I have to recall that we have studied in our earlier class with respect to the restriction enzymes. So we also have what is known as an isoschisomers similar to isoenzymes. So we do study isoschisomers like as either isoschisomers or neo-isoschisomers or isocardamers. I will just define what are they. So isoschisomers are nothing but two different restriction enzymes isolated from two different microorganisms. Independently, these two restriction enzymes recognizes the same restriction sequence and also initiates digestion of this restriction sequence in the same site. So Hindi3 recognizes AAGCTT, HSU also recognizes the same sequence, and both of them cut the phosphodiester bond between AA and results in the generation of a sticky end. They are called as an isoschisomers. Whereas we have neo-isoschisomers. Here we do have two restriction enzymes isolated from two different microorganisms. Both of them recognizes the same restriction sequence. Compared to this, they initiate cutting at different place. Small one recognizes the same sequence, but it cleaves the phosphodiester bond between C and G that results in the generation of a blunt end. Whereas XMA1 recognizes the same sequence CCCGGG but initiates the cleavage in a different site between C and C. Now this results in the generation of a sticky end. Now we have a third format, what they call this as an isocardamers. So isocardamers, for example, BAMH1, BAGEL2, BCL1, all of them are isocardamers. BAMH1 recognizes a different sequence, BAGEL2 recognizes a different sequence, BCL1 recognizes a different sequence. They are all different restriction enzymes isolated from different microorganisms. They recognizes different sequence, but the cleavage product are compatible. For example, BAMH1 cleaves this particular site and results in the generation of CTAG as a overhang. BAGEL2 recognizes totally a different sequence, but thus cleaving product is again CTAG. Now this and this are compatible. Whereas BCL1 again recognizes a different sequence, they generate CTAG. Now the other counterpart is CTAG. So now suppose like if there is a site BAMH1 generate CTAG, BAGEL2 generate CTAG. Now BAGEL2 site and BAMH1 site can be used for cloning purpose. They are called as an isocardamers. I told you BAMH1 is extensively used in cosmic vectors. Now this BAMH1 has got a compatible end produced by SAU3A as well. Okay. Now the difference is that BAMH1 is it recognizes only 8 base pair, and that is quite frequently this is available. Alright. So as a result, BAMH1 is not used to cleave the eukaryotic DNA because it will generate lot of smaller fragments. So because of that, BAMH1 is not used to cleave the eukaryotic DNA. So instead we use a rare restriction enzyme SAU3A, which their sequence is quite rare, but once they do generate fragments, they also produce sticky end, but that sticky end is compatible to BAMH1. So this particular strategy is very, very important. Alright, to begin with, so what do we do now? We set up one reaction. So we take the plasmid and we digest with Hindi3. So now Hindi3 cleaves here. So now that is the what we are going to generate is going to be that, okay, now there is a linearized plasmid, we have a COS site, and now there is a BAMH1 site. So now this is in one reaction. Again we are going to set up a second reaction separately. So now you take the plasmid, so we will digest that with SAL1 enzyme. Alright, again that results in the linearization of the cosmid vector. So now here is a cos, and now here is going to be the BAM-H1 site. Now what we do that, so in order to avoid the tandem repeat, or what you call the linearized polymerization, so we treat both the DNA fragments with phosphatase. So now we'll take this one treated with phosphatase, we take this DNA treated with phosphatase. So what this phosphatase does, it removes the phosphate from the 5' region from this end, this end, this end, this end and this end. Now what we are going to generate is going to be that both the end has got hydroxyl, there is no phosphate. So in the absence of phosphate, there is no way that these two can combine together during the ligation process. It is not going to be possible. So that is the very first strategy. Now, in order to make this one for cloning purpose, what happens, like now we take this digested with BAM-H1. So now what happens, now this particular end is going to be cleaved. So now we are going to generate a sticky end with BAM-H1 protruding. So now I will just written like GGATCC. Now this cuts here. Now the product what we are going to get CTAG. Now there is a OH. So now here we have both OH on either side. So because of the cleavage, so we have generated a sticky end and also we have a phosphate here. And the same is true here. So now we have digested with this. So we have the GATCCG. So now this end and this end are compatible. So now there is a phosphate group here and there is a phosphate group here. So what happens? Now this end is going to be completely hydroxyl. There is no way this end and this end can combine together to form the ligation product. It's not going to be possible. So the only way it is going to be possible is between this end and this end. So now you take this cosmid linearized vector treated, and this you take it up, and with this particular fragment, then we are going to do the ligation. So we need this fragment, we need this fragment, and we need this fragment. We combine together and we start the ligation process, in fact. Now two things can happen. This end and this end can combine together to generate a linearized DNA. So we have two cos sites. Now the distance between two cos sites is so small, lesser than 38 kilobase, which is required. This is not going to get packaged. So whatever the number of ligation product you are going to get this particular species will never get packaged. Alright. Now the other one is going to be that now this is one end, this is another end. In between we have the DNA which is going to be inserted. Now this is the right clone. Now there is a cos site, there is a cos site. Because of this, now the distance what is required between 38 to 50 is generated, and this automatically get packaged in fact. So now this is a wonderful strategy, and so now the use of the restriction enzymes, phosphatase, and later treated with BAM-H1 to generate the required site. And we have to understand, like BAM-H1 has got a compatible site with SAU3A, which generates larger DNA fragments. So these are all the selection procedures they have used to generate this particular clone. So now what happens, like you take this clone, do an in vitro packaging, then after that again you try to infect with the regular E. coli so that it will enter into the lytic cycle. So I will tell you another strategy using another vector. Okay, so I'm going to use this portion same for this vector. So now here this vector is C2XB. So now the size is 6.8 kilobase. So now here this is a circular plasmid with ampicillin resistant gene. So now here it has got two cos site. Alright. So now in these two cos site, strategically we have a SMA1 and another one is going to be the BAM-H1. So the SMA1 is placed in between two cos site. So unlike earlier, we take this particular plasmid and we digest with SMA1 and BAM-H1. And we have to know that BAM-H1 digests and creates a blunt end, and BAM-H1 creates a sticky end. So now this is one fragment BAM-H1, and the small one is going to be here, and this is the BAM-H1 and then the small one here. Suppose during the ligation process, it is very much clear that sticky end ligation is much more preferred than blunt end ligation. Suppose you two take these two things, the joining of this end is more preferred rather than joining of this end and this end. So as a result, so now in the previous one we treated phosphatase followed by treating with BAM-H1, but here we are avoiding that basically because we are digesting with sticky end and a blunt end with an intention that blunt end is not preferred for ligation when compared to the sticky end. So now we take these two fragments, then like how I discussed in the previous one, we digest with SAV3A and we are going to get these fragments, these two fragments and these fragments, we make the ligation. Alright, so in case of a ligation, there is a chance that this end and this end can combine together to form this species. Obviously, this is less than 38 kilobase, it will never get packaged in. Whereas this particular end, and this is another cosmid vector, this is another cosmid vector. Now, this is the BAM-H1 and we have a compatible BAM-H1. Now, this is going to be ligated. Now, since we have the two cos site and that distance is, whatever the distance is required by this 38 to 50, if it is maintained, this is automatically get packaged in fact. So, these are the strategies employed for cloning larger DNA fragments with the cosmid vectors in fact. Now this completes a major portion of vectors. I have discussed plasmid vectors, we have studied in detail till the somatostatin hormone insertion, getting a clone of that, expression of that, measurement of that. So we have really understood that, and also how PBR322 plasmid vector has been constructed. So for then plasmid vectors has a limitation. To overcome that, we discussed about the Forge vectors. In order to understand the Forge vectors, we discussed in detail about the lysogeny cycle, the lytic cycle, the important promoters which are used, and all those characteristics I think we have studied. Using that, I think we have Forge vectors either insertional type or a replacement type. We have the beta-gal gene is very important, C1 repressor gene is important, red and GAM gene is important. So with that we can generate a linear DNA. So in that forged DNA, I think we can insert 24.6 kilobases. But for the insertions, we have to discuss about the in vitro packaging. We also discussed about what exactly is an in vitro packaging. This usually takes place in a mixture of lysate of a mutated strain. And with that, once you understood the in vitro packaging, we also studied what is very critical. The only critical thing is going to be the two cos site and the distance between the two cos site. So as a result, we have generated the cosmid vectors. So in case of a Forge vector, we have 24.6 kilobase maximum can be inserted, whereas in case of a cosmid vector, even larger than that can be inserted. So stepwise, plasmid is started with that, then the Forge vector, then the cosmid vectors. So in case of a cosmid vectors, in vitro packaging followed by the infection is very, very critical. In case of a plasmid, we generate colonies, but in case of a Forge vector as well as cosmid vectors, so we are going to, if it gets us into the lytic cell, so we are going to get a very transparent colony, what we call this as a plaque formation in fact. That completes everything, a major portions of recombinant DNA technology. I hope you have understood everything. Thank you very much.